Good afternoon, everyone, and welcome to this special session on the COVID update as part of the National Academy of Sciences annual meeting. I'm Victor Dzau, the President of the National Academy of Medicine. I want to begin by thanking Marshall McNutt for inviting me and our panelists to engage in this really important topic. And so today, what I'd like to do is begin with just a few opening remarks, tee up the issue for the day, and then turn to our panelists, who are the world's expert in this area. And of course, you want to hear from them, not from me. So since January, the world has been facing a global crisis, the COVID-19 pandemic. This pandemic caused by coronavirus SARS-CoV-2 is having a devastating effect on the world in terms of life lost, human suffering. It impacts our communities, people's lives, hurts the economy, and the crisis is truly affecting everyone. As of today, a few minutes ago, these numbers keep changing, but globally, we're seeing about 2.85 million cases and close to 200,000 deaths. US is taking the major brunt. We have 925,000 cases and 52,000 deaths. So you can see why, certainly in our country, and globally as well, this is such an important issue. Now, although a lot has been done, and we are making progress, but there's so many uncertainties, and there's so much more work to be done. So today I'm joined by an outstanding panel of the world's leading expert in the area of global health, infectious disease, virology, vaccine development, clinical medicine, you name it. Now, I must admit, I got a little bit carried away. I got a little greedy. I invited 6 exceptional leaders, my good friends. To my dismay, they all said yes. So you can imagine how important this meeting is. So here we are, and we have 90 minutes, and I will introduce them very shortly, but let me tee up the agenda for the panel. Can I have the first slide, please? Or next slide. So these are the questions. 1, what do we know about the virus? And of course, what we don't know. 2nd, what's the state of pandemic? Where are we in the US? And have we passed the peak? And where are we in different parts of the world? Here we don't hear a lot about Africa, but the South could be the next wave of this infections or outbreak. So what do we know about this? How effective is mitigation versus containment strategy? We hear from speakers from China, United States, UK, about the success of relative strategies, and of course, the experience of these countries. What's the current state of testing? This is a big deal in the United States. You know, there are 2 types of testing, molecular testing and serological testing. You're going to hear about that. But what we know is that testing is sorely needed to understand where we are and how we're doing in terms of the outbreak. Testing is needed to inform the state of immunity of the population and decisions about when to relax some of these measures. Well, what about therapeutics? We certainly need therapeutics. There are 7 drugs now in almost 300 clinical trials, to my knowledge. Some of them are quite well known, like hydroxychloroquine, but also the antivirals, like remdesivir, and of course, convalescent plasma. Vaccine. I think we all recognize that the only way to ultimately stop this virus from spreading is vaccine, and to prevent future resurgence. So question is, where are we in vaccine development? What are the technology platforms? How many vaccines are out there? And how soon will it entering clinical trials, and how soon will we know the result? I think also in discussion by at least 2 or 3 of our speakers is the coordinated effort globally to accelerate diagnostic vaccine therapeutics, not only in terms of research development, but manufacturing, distribution, and how to ensure equitable access. Impact on vulnerable population and the whole issue of equity. Certainly we see starkly in the United States this issue of social inequity affecting Latinos, African Americans more. Of course, we know the elderly. We'll talk about that too. And of course, technology. There's a lot of innovation out there. I'd love to hear some of my panelists thinking about the use of digital technology, tracking people's symptoms, contact tracing, and measuring the effect of social distancing. I'd like to end by saying, have we learned anything? This is not the first time the world has experienced an infectious outbreak. We had Ebola not too long ago. We have SARS. We have many others. And many people have written about what are the lessons learned, what needs to be done. Are we finally going to wake up and learn and take action to ensure preparedness for the future? And what would our society look in a post-COVID world when every part of society has been affected, every sector? When we emerge from this pandemic, we need to design and build a more robust, resilient society with stronger economy and rebuild our infrastructure base. So with that introduction, let me welcome our outstanding panelists. First is Jeremy Farrar, who's Director of Wellcome Trust. Jeremy, welcome. Tony Fauci, need I say more? Director of National Institute of Allergy and Infectious Disease. Sanjay Gupta, need I introduce him? A neurosurgeon, a chief medical correspondent of CNN. George Gao, this is now 2 a.m. in Beijing. And George been willing, when I sent him the note, I thought he would say no. He said yes, so there you go. So George, I'm sorry to stay up a little bit longer later, but he's the Director General of China CDC. Susan Weiss, Professor of Microbiology at University of Pennsylvania School of Medicine, and Richard Hatchett, the CEO of Coalition for Epidemic Preparedness Innovations. So welcome all of you, welcome. Let me start, therefore, each panelist will get 7 minutes. Some have slides, some do not, but all told, they're going to tell you the story and, from their perspective, but then should have about 30 to 40 minutes of Q&A. Start with Jeremy, my good friend, Director of Wellcome Trust. Jeremy, you're truly a leader in global health. You chair the WHO Blueprint. You're a member of the Global Preparedness Monitoring Board. Tell us how you see the global state of this pandemic. Where were we? Where are we? And what do you see ahead? Jeremy?
Victor, thanks very much. I hope you can hear me, and it's a huge honor to join the National Academies in this and be with such long-standing friends on the panel and such prestigious colleagues. I do think we've got to remind ourselves we are only about 120 days since the world learned of the emergence of this new virus, a brand new human infection which within a month had spread intercontinentally and has now affected essentially every country on Earth. And yet at a time when cities and countries, including the United States, is suffering enormously from this pandemic, I do have a sense that we still as yet underestimate the true impact of what we face today and what we may face in the coming months and years. This is clearly a health issue predominantly, but it is also a social issue, an economic issue, and indeed a political issue. And we have to address all of those as we both go through and hopefully at some point come out of the current situation. And just in starting, I'd like to pay tribute to healthcare workers all over the world who are putting their own lives at risk so that the rest of us can be that little bit safer. The cases numbers, Victor, you've just mentioned, 3 million cases roughly in the world and 200,000 or so tragic deaths. The US, I know, has just passed 50,000 deaths. The UK has just passed 20,000 deaths. Horrendous numbers for something that we didn't even know about 3 months ago. But the true numbers are going to be hugely more than that. Recent data from London suggests that between 10 and 14% of Londoners in the middle of March were infected with this virus. So the true numbers globally are going to be very much more. And the true numbers of deaths in hospitals, UK 20,000 this week, do not reflect the total number of deaths in the community from COVID, let alone from the other conditions that has made, because of the difficulties in running a health system in the middle of a crisis like this, inevitably the number of deaths occurring from other conditions where the health system is under such threat. So the numbers are significantly more and will continue to rise in the weeks and months ahead. This is an RNA virus, as we'll hear later, originally linked to bats, but clearly there is another intermediate host between the bat and the humans, not yet identified. The virus spreads by the respiratory route. It is transmitted from the pre-symptomatic phase right the way through until people are in hospital. So the period that people remain infectious is very long, and you have very high viral loads in both the nose and throat, increasing the capacity to spread this to other people. The R0, the number of people I would infect today if I were infected, ranges from somewhere between 2.5 and 5 when the pandemic has been at its peak. That's an incredibly infectious virus. And this is the sort of infection that many of us, everybody on this panel, has been warning the world about for the best part of 20 years. An animal virus that comes across into humans, to which humanity has no immunity, which has the ability to go human to human with a high rate of infectivity, spread by the respiratory route, and for which there is a very broad range of clinical syndromes, from asymptomatic all the way through to the need for intensive care, and of course, tragically, a very large number of deaths. And an infection for which we had no diagnostics at the start, no treatment, and no vaccines. Clearly, Europe and North America is now the epicenter of this pandemic, but my biggest worries next are going to be what happens when this increasingly becomes an issue in Central and South America, in Africa, and South Asia. And indeed, in those communities, as well as in North America and Europe, the impact on marginalized and vulnerable populations will inevitably be bigger. Pandemics always have an unequal distribution, both in society and globally. People in marginalized populations in America and in Europe will continue to suffer longer than those potentially in the big urban centers. Rural communities will be particularly hard hit when the infection reaches and takes hold in those communities. Around the world, conflict zones, refugee camps, migrant camps, and informal settlements will prove to be hotbeds of this epidemic. In the UK and in Europe now, mostly the R0 has dropped below 1, and therefore the epidemic is starting to plateau in many parts of Europe. But that has only come about as a result of draconian social distancing measures, which have brought economies to a standstill, and which in themselves have hugely damaging impacts both on people's health, people's well-being, people's mental health, and other conditions beyond COVID. The lockdowns cannot go on forever. The lockdowns buy us time, they reduce the peak, they reduce the R0, but in themselves, they are not an exit strategy. An exit strategy can only ultimately come from a massive investment at a global level in science, and not just the science, but also the ability to deliver that science in an equitable way to to everybody in the world, independent of their ability to pay. Because as always, whenever you have an epidemic of this scale, it isn't just the infection that matters. It's the infection itself, COVID-19. It's the broader health issues that therefore get often neglected. It is the societal issues and the inequalities that come from it, and inevitably tensions that can arise across borders, which we must do all we can to reduce. And this needs massive investment in basic science, so we understand the virus and the immune response, in social sciences, so we understand the behavioral aspects, and of course in the development of diagnostics, treatment, and vaccines. But that science on its own is not enough. Unless we commit ourselves as a world to making sure that science is available to everybody, the vaccines and the treatments are manufactured and distributed independent of people's ability to pay, then we will not bring this pandemic to an end. That is both enlightened self-interest and it's a global public good. And it's something I believe we are all and must be committed to. In addition, we must start to look, even as we're dealing with the crisis, we must lift our eyes to deal with what we know is coming, which will complicate matters even further. As the Northern Hemisphere goes into its flu season, we will have the double whammy of this COVID potentially returning in second and third waves, plus influenza season in other parts of the world, malaria seasons, dengue seasons, and other comorbidities which will complicate how we address these issues. So through science, I believe we can exit this, and if we commit ourselves to making sure that science is shared to the benefit of everybody, I think we can actually come out of this in a better world than we went into it. Throughout history, the world has faced great crises. Never before, however, have we all faced it together in an equal way. In the aftermath of such crisis, there is always a choice which societies have to make. Do we seek to apportion blame, exact reparations, and become ever more polarized? Or do we seek to come together to learn the lessons that caused the pandemic, to learn those lessons and refashion a more collective, a more equitable, and a more cohesive world to address our shared challenges through the 21st century? We as a generation will face such a choice, and because on the basis of the choices we make, will define the 21st century. Victor, thank you very much. Hand back to you and my colleagues on the panel.
Thank you, Jeremy. Inspiring words. It's a perfect setup for the rest of the discussion. I would say personally, it's a great pleasure working so close with you in many different issues. My great admiration. Thank you. Let me turn to Tony. Tony, you're seen as one of America's most trusted medical figures. You served 6 US presidents, and you're a fixture on CNN and other news sites. I heard that you even spoke with Stephen Curry, one of my heroes on Instagram. So I know you, and because we all know you play a major role in the White House task force, and you're a trusted voice of science to the public throughout all this. So tell us about US, and of course, the great work you and others are doing, but tell us what to expect, and are we out of the woods?
All right, thank you very much, Victor, and thank you for giving me and my colleagues the opportunity to speak with you today about this extraordinary, difficult situation that we find ourselves. Jeremy did his usual spectacular job of introducing and expressing, I think, the feelings of most, if not all of us, who are involved with this, and gave a very nice global perspective. I'm going to talk, as you mentioned, about the situation here in the United States. As Jeremy said correctly, the United States is now the country that has suffered most. That does not mean that there is the worst ahead for many other countries, particularly in the Southern Hemisphere. As you can see from this slide, we have almost 1 million reported cases and over 50,000 reported deaths. The size of the circles throughout the country indicates something that's very clear, that New York metropolitan area has really suffered more than any individual city, as you can see here from the number of cases and a proportionate number of deaths. As you know, the outbreak in the United States likely started in the state of Washington and in the state of California, and then worked its way rapidly across the country. That came from China. We now know from sequencing data that it is highly likely that the New York cases originated from Europe and not from China, as we're starting to get a better feel for the different subtypes, or if you want to call it mutational changes in the virus. I'm sure you'll hear more about from Dr. Weiss. We have a number of cities which have peaked, and some of them are not yet peaked. New York City, as I'll get to in a moment, has actually had a very interesting situation with regard to the number of deaths had continued to increase at the time that the number of cases and hospitalizations were being stabilizing and coming down. So if you look at the reported cases per day in the United States and the reported deaths per day, you can note that we in the United States, with these interesting little intermittent blips, have reached a point where we are starting to see a stabilization. But this is misleading. And I want to get back to the situation in New York metropolitan area, because this is such a large country and the dynamics of the outbreak differ from state to state, from region to region, and even from city to city. So at a time where you may see New York City peaking and coming down, it gets confusing, because on the same week that they had the worst deaths that they've had, we were starting to see a diminution in cases, hospitalizations, requirements for intensive care, and intubations. So the deaths always lag. So when you look at statistics, you may see something where a city is starting to stabilize, but the deaths, which are 2.5 or more weeks following the actual identification of a case, that's when you see what looks like a disparity. So these are the numbers here that we've seen. We can get back to some of that in the questions later. We have a very interesting situation in the United States, a major disparity, both in the risk of getting infected, the risk of getting hospitalized, and the risk of dying if you compare African Americans, which comprise about 13% of the United States population, compared to Hispanic Latinas, whites, and Asians, the data are very striking, as you can see on the current slide. The test in the United States, I mean, obviously is very clear to everyone because of the publicity that it has gotten, that we got off to a difficult start with regard to testing. As you can see, if you go back to March 15th, and then go up to where we are today with about 5 million tests that are being done. I think we could have a whole discussion, which I would certainly welcome later on in the questions, about testing, but clearly we are much better off today than we were last week, and we will be much better off a week or 2 or 3 from now, as now we have the capability with the private sector getting involved in the making and the distribution of tests and the proper capacities being utilized. Again, a very complicated problem that has gotten a lot of attention. The NIAID, which is the leading institute within NIH, but there will be other institutes involved, as we'll get into, I hope, in a moment, has developed a strategic plan looking at fundamental knowledge of this. You're going to hear about that from Dr. Weiss, but there are so many things about the virus and the pathogenesis that we do not know. And literally every day and week that goes by, we learn more and more. We need better diagnostics and assays. We need to characterize and test therapeutics. And as you've heard from Victor and from Jeremy, safe and effective vaccines are going to be the bottom line of how we end this. With regard to therapeutics, there are many, many that are being now introduced into either preclinical or clinical testing. These are some of the ones that have been in clinical testing and investigating. Everyone knows about remdesivir. I'll get back to that in a moment. Other broad spectrum antivirals that are now being repurposed, convalescent plasma and hyperimmune globulin, repurposed drugs like hydroxychloroquine and lopinavir/ritonavir, immune-based therapies blocking IL-6 and IL-6 receptors, monoclonal antibodies, and others. Remdesivir is the one that's gotten the most publicity. It is interesting that if you look at it in a prophylactic therapeutic model in MERS covirus infection, it looked pretty good. There was clinical benefits just reported a couple of weeks ago of SARS coronavirus 2 in rhesus macaques. And we have a clinical trial now, a large clinical trial that is going to answer the question. We've heard already about some inkling that this is not going to be a knockout drug. You never want to get ahead of yourself in a clinical trial, but this was the first clinical trial to evaluate this. The participants are Americans who repatriated after being quarantined, and we have studies can be adapted to evaluate additional investigative treatments and participants at other sites. Now this adaptive trial that's randomized goes literally throughout the country and the world, and we will get an answer. I want to take this opportunity to emphasize something that I hope we can get a chance to emphasize even more later, and that is the only way that you're going to get an answer that is not just perpetual ambiguity is by doing a randomized controlled trial, because as you can see, there are so many claims in there, and I'll get back to how we're trying to get around that this works or doesn't work. I mean, we need something out there, but safety and efficacy is something we owe to the global population. And then finally, with regard to vaccines, these are a number of the platforms. There are at least 3 that are already in clinical trial in the United States, and I'm sure globally there are several others. We're using everything from genetic immunization with RNA and DNA vaccines, viral vectors, live attenuated proteins, nanoparticles, etc. I'm not going to spend any time on this key spike protein, which is the target of a vaccine-induced antibody, because I'm sure you'll hear about that from Dr. Weiss. We have a clinical trial of investigative vaccine that we began in phase 1 several weeks ago. First is 45 healthy individuals. We hope to get into phase 2 in the late spring and early summer. We could talk a little bit later about what it's going to take to ultimately get an efficacy signal. Obviously, you're going to be in a situation where you have active ongoing cases. If you look at the time frame of vaccines that we have developed, genetic vaccines starting with the SARS DNA protein, it took 20 months to go from sequence to the first phase 1 trial. And if you look how we diminished that requirement with H5N1, H1N1, Zika, and now coronavirus, it was actually 62 days from the time of sequence. And that's the one that's right now, the mRNA, already well into phase 1 trial. But this is not the only one. We could talk about the others. I think this is very important, given the fact that there are so many claims, anecdotal, about what works and doesn't work, that we put together at the NIH and we put it public about 4 days ago, on Tuesday, April 21st, a guidelines that are now online in the living document. We brought together people with extensive experience from all over the country, multiple federal agencies involved, led by the NIH, chaired by Cliff Lane, Henry Mazur, and Trip Gulick from Weill Cornell Medicine Presbyterian. And right now it's available to anyone. And I just took a look at the number of hits it's got. It's almost 1 million page hits, and over 60,000 individuals access this. As you know, we have a coronavirus task force, of which I'm a member, and these are the individuals in that task force. We meet every day, 7 days a week, often into the evening and night, which some of you have seen because there's usually a press conference after that. I want to close by just spending a minute, and I'm sorry, I think I'm a minute or two over. I'm sorry, Victor, but I think this is important to bring up something that is going to be very important because of the devastating economic impact that this virus is having throughout the world, certainly in the United States. We have put together a program that's a careful, well thought out program of slowly and carefully going into a program of trying to reduce the mitigation practices slightly, step by step. Mitigation that we did by essentially shutting down the country for the most part was the reason why we're turning the corner. I have warned the public, and I'll do it here, that the process of getting back is a multi-step process in which you must pass certain step points, which we call the gateway, to go into phase 1, phase 2, and then phase 3. Any attempt to leapfrog over these almost certainly will result in a rebound, and then we could set ourselves back. I'm sure that we're going to have a lot of discussion about that, but I thought it would be very important for me to emphasize that up front, because you hear a lot about the need and the desire to get back to normal. That's understandable, but we're dealing, as Jeremy said, with an unprecedented global health problem. If we don't get control of it, we will never get back to normal. I know we will, but we've got to do it correctly. I'll stop there. Thank you.
I thought your presentation was comprehensive, thorough, and full of information, and thank you for your leadership. We'll come back to you. Let's turn to Sanjay Gupta. Sanjay, everybody knows you as a great science communicator, but you're also a neurosurgeon. I know you're in the front line, and you talk to a lot of people in the front line. And so being a clinician, communicator, tell us about the U.S. health system response. What are the frontline workers dealing with? What about the issue of supplies? And what have we learned about our system and what needs to change?
Yeah, well, let me just say I'm very honored to be a part of this, first of all. I mean, this is an amazing panel and people that we should be hearing from. So thank you very much. I am already getting a lot out of this. And let me also single out Tony Fauci as well. I mean, as you pointed out, Dr. Dzau, he is the most trusted man, I think, in America. And I think it's not just his knowledge, but his humanity and his diligence as well. So thank you for that. And thank you for inviting me today. I'm really, really humbled. I thought I'd give some remarks along those points you're raising. And I might start just by just giving you a few thoughts from my vantage point as a science-based journalist as well, Dr. Dzau. You know, it's been a really interesting time for us. I've been doing this sort of straddled career for some 20 years now, and this has been a wholly unique situation, obviously, for everybody, including us in the world of journalism. Our thoughts and our insights and our reporting comes from evidence and data and less opinion and conjecture. And I realize and have realized for some time that is a real luxury to be able to be in that part of reporting. It does require a lot of reading, constant reading in a variety of fields, oftentimes going back to the basics. I've probably read more of Harrison's Principles of Internal Medicine now as a medical journalist than I did as a medical student. Of course, I read Dr. Fauci's chapter again as well. But I also have on my daily reading list lots of journals, lots of newspapers. The journals are both published and preprint journals. We have to keep up. And there's an incredible appetite for this knowledge from people all over the world. And we are a global news network. I think one of the principles that I've let guide me for the last 20 years is that I really need to feel like I understand something thoroughly before I start talking about it in any kind of public setting. Now, perhaps that sounds kind of obvious, but I think the complete level of learning and understanding required now is higher than ever. What has really struck me is that the entire world is watching and listening and questioning every single thing. And it does expose any weaknesses or even slight inconsistencies in how we talk about these things. It exposes the gray areas and the nuances. And I think in many ways it lays bare the part of the conversation that we doctors always have with patients, where at some point, you've explained it the best you can, and you're at that point in the conversation where you're sort of saying, hey, look, part of this is going to be that you have to trust me. You do have to trust me a little bit on this. And it's challenging to do that when you're speaking to a global audience. Again, no one knows this better than Tony Fauci. As a doctor or a journalist, again, straddling these worlds, it means, really locking down all the existing knowledge that you can have, begin to anticipate and explain inconsistencies in a way that makes sense to you first and then to your audience, whether it be a single patient or, a television audience. And with the story of COVID, I think that this part of it is probably the most challenging that I think I've ever experienced. We have not been able to incorporate, I think, what the audience typically wants of us, which is to say, based on 10 years of data or based on 20 years of data, sometimes we can't even say that in weeks or in days. So I think that there's many things that, we have been focused on, try and focus on all the threads of the story, but some obviously more than others. I think that the origin story, which has already been alluded to, is something that keeps coming up over and over again and does need to be analyzed more completely and retrospectively. It's something that the audience really wants to know. Perhaps it's not as important as what is going forward, what is more prospective, but it is something that comes out. And that part of the story has been opaque. And as a journalist, it's been really difficult because of that opaqueness. It's hard to be reassuring sometimes that the original infections seem to originate the way that they did when we subsequently learned that the first confirmed patient, for example, had no interaction with that animal market that was talked about a lot in the news. In fact, a third of the patients roughly that were in the original paper did not have contact with that animal market. When it is presented as this is the way it is, this is an animal to human virus, and they all came from this animal market, which was some of the initial information we were getting, and that subsequently turns out not to be true. It is a challenge then as a journalist to keep presenting this information because it throws into question many, many things. There's a lot of lessons, and I thought I might just give a few from my vantage point. Again, some of them have already been alluded to, but I think the most important one is the need for humility as we talk about this. We do present stories, based on science and based on data and based on evidence, and I think everyone here would agree with me that is often thought of as a 100% proposition then. Here is the data, here is the science, and that is a 100% proposition. There's 100% certainty behind it because it has data. That is often the interpretation from the public. But I don't think I'm out of line to suggest that there are very few things in life that are going to meet that standard. And with this particular story, it is again very much the case. We've been reminded how important it is not to be overly dogmatic, I think, about these things, and to constantly use the phrase, based on what we know now, and to be humble about it. In the beginning, I think, as I mentioned, a lot of journals had to dust off their previous epidemic and pandemic reporting. We did a lot of reporting on SARS. We did a lot of reporting on H1N1. And I think in many ways that was the framing in which this story was originally presented, certainly by science-based journalists, because that was the context that they had. Was this going to be more like one or the other, SARS or H1N1, or was it going to be like neither? That was sort of the way that it was presented often. And by the way, also recognizing that since the last pandemic of H1N1, that the world had changed a lot. And how to frame that as well. Global travel roughly has doubled. There are roughly twice as many people who now make international flights now versus when they did during the 2009 H1N1 pandemic. It's also important to remember, as we try to do as medical journalists a lot, and I think we all do as clinicians, is to remember not to use the words like cases too often. These are people. They have confirmed infections, and it sounds sometimes less than humble and diminishes the gravity of what we're speaking when we use words like cases. It starts to sound like an amalgamation of numbers, like a numbers sort of thing rather than real lives. And again, we all recognize that, but sometimes the language can lapse. And I know it's not intentional, but especially for people who are watching, increasing number of whom every single day are directly affected by this, myself included, in terms of having lost friends who have died of the COVID infection. It's something that we really try and remind all of ourselves, certainly in the world of media. I think one of the other things in terms of lessons learned is this idea of how best to present risk, how best to present the concept of risk to people. So many times equivalence gets drawn between something like this, coronavirus, and car accidents, for example. And it's a false equivalence. I think everyone here would agree, and I know Dr. Fauci has been asked about this and has said, but we need to explain why that is. Why is it a false equivalence? Because, again, too often just the numbers are presented. I'm looking at these numbers and I'm looking at those numbers, and these numbers are actually higher. So why are you making such a big fuss about these numbers? That one has no protection. That one is the risk factor can be as simple as breathing or touching your face, and that one can grow exponentially. And then explaining what exponential growth really means. Something that sort of starts more slowly but then grows rapidly. How do you distinguish that from logarithmic growth, for example? And the audience cares about this sort of stuff. It's very interesting. I make this point, and I'll just finish with this because I know I've already gone over my time, is that I think it's important to appeal to the highest common denominator when talking about these stories, these important concepts on television. I think a lot of times the fail-safe is to say, let's make this as widely palatable to the largest number of people. And I get that, but I think now is the time to sort of raise the denominator as opposed to appealing to the lowest common denominator. There's a real sense, that as we talk about this more and more in the days and weeks ahead, that how we talk about it will be just as important as what we say. I think that this is something, again, everybody knows is affecting the whole world, and I've never seen the appetite for knowledge around this as I've seen with this. Dr. Dzau, I've already gone over my time. I'll talk about some of those other issues maybe in the Q&A, but again, I'm humbled. Thank you very much for having me.
Thank you. And Sanjay, I think you have just kind of provide strong evidence why science needs great science journalism, and the public needs great science journalists, so that we can really bring science and its understanding to the public and to ourselves, that we can communicate the work that we do to the public. So let me turn to George Gao. George, now it's 2:30 in the morning, and drink a little more coffee, George. But we know George well because George actually has done a lot of work with national academies. I think he was involved with our Human Genome Project and a couple of other editing project and others. But George, as the head of the China CDC, and China was the first to experienced the disease, and your country quickly implemented strong measures in Wuhan and also in Hubei province. As one of the audience just sent me a note to say, with 1.4 billion people, having 82,000 cases, or pardon me, Sanjay, of people infected, that's quite amazing. And now you're lifting restrictions. So tell us about the strategy, lessons learned, and then how are you going to be sure that you don't get another wave of infection? George, over to you.
Hi, Victor. It's a great pleasure for me to be here. First of all, I want to also pay a tribute to all those heroes who are still working in the field against this COVID-19. And also I want to pay a condolence for those who sacrificed their life in the world fighting against this COVID-19, especially someone who sacrificed life in Wuhan. That's the initial place we discovered the COVID-19 disease. First, I show you this slide. So this slide, within a week, once we noticed there is something there, within a week, and China CDC, in collaboration with 3 other labs, we worked together, we isolated the virus, we did all the whole genome sequencing, we know what it is, we shared the sequence data, and we developed the diagnostic kits, just within 10 days, we got everything. I know this is a totally different experience when you compare with SARS. This is a definitely science-based identification and science-based control in China. Victor, that's a very good question. Whenever I show the slide, share with a lot of my colleagues from all over the world, people always ask me the question, we almost 1.4 billion people, and why you didn't have so many cases? I will show you. Soon, Dr. Weiss will give you maybe detail for virology. I'm a virologist, maybe I can give for that, but instead I'll tell you what we have done and where we are in China. So next slide, please. Yeah, this is, look at this. We noticed this by the end of last year, 31st, retrospectively, when we move back, this is what everything is reported in our the end of January paper in New England Journal of Medicine. Retrospectively, we might have some sporadic cases, during December, just because during December and January, that's the flu season. You have a lot of cold, flu, all those places. But you look at this, and all the places they started roughly, reach to their peak about 23rd, 24th of January. But look at the smaller one, the red one. This red one is tells you, all the peak wave 1 got down, but now we have the problem with the imported cases. That's also a very good question. Victor, you asked me, are we expecting the second wave? Well, I think we have to wait and see. So we are working very hard. I'll show you what we are doing at the moment later. And also surprisingly, look at this figure. We have one column, it's very high. I saw this figure also from Tony's, in the United States. You can see during that time from very beginning, you have a lot of things. And, when you calculate and why you have so many patients, especially recently, we readjust the death patients and the death numbers. People ask, why you have this? I mean, the same thing like what Tony showed, because at the very beginning, it's not really, well ordered. You have a lot of things like that. So next slide, please. So this is the lessons we learned, in China side. We call this one first, everything done by science-based or science-driven. And also what we did, because we don't have the vaccine like Jerry mentioned, and we don't have any specific drugs. So what you can do, only the traditional method. So this is so-called non-pharmaceutical intervention, OPI. We use all this OPI and also community mobilization. And also in China, we try to block the public transport. Of course, I don't think, a lot of countries you can't learn from us. I'm not telling you should learn from this, but I'm telling you what we have done, what we did, in China. And also, all this, you need a very strong leadership and coordination and cooperation, because China, not only with the 1.4 billion people, but also, geographically, it's a huge country. So you think, when you talk to the people in different regions, you need a very good coordination. You lock down Wuhan and Hubei province, and how would you coordinate well with other regions? So this is, something, from the very beginning, we did, especially for the logistic support. All these need a coordination and strong leadership. This is what we have done. Next slide, please. Very important, from the very beginning, we realized, you cannot have one strategy, you cannot have one plan, you would solve all these problems. So this is why we call it the science-driven, timely adjustment. We always adjustment and readjustment. This is why we try to categorize the regions with low-risk area, middle-risk area, and high-risk areas. We try to categorize the regions by, define them into those three different areas, and we also should readjust the strategies and programs and the plan, for this. I don't want to go into this in detail because of time limit, but our next slide, please. For that, even for the diagnostic and treatment protocols, prevention and control protocols, we readjust from version to version. Now we have, already version 7, and we are preparing the version 8. You have to readjust, retuning whatever the protocol you have. Next slide, please. Here's the so-called lockdown or containment strategy in China, what we have done. First, of course, everybody is talking about social distancing. So avoiding crowding and extending holiday, because from the very beginning, that's the time for the Chinese New Year. It's very hard for us. So this is why we had the lockdown. And then you have to have the detection, isolation, quarantine, early detection, early report, early isolation, and early treatment. Those 4 early is very, very important. And also the movement restriction, suspending the public transport for Wuhan, entry and exit screening to Wuhan. This is a so-called lockdown. And also infection prevention, especially the nosocomial infection, and environmental measures, especially disinfecting the initial place in the market, and also social mobilization, health education, and risk communication. Here I want to address risk communication, because of the word infodemic. We pay a lot of time to do this, try to make sure the public understanding what we are doing. Public understanding and public involvement is very important. As I said from earlier, science-based measures, but whatever you are doing, you have to make sure public understand what you are doing. You have to make sure whatever strategy you impose, the public will be involved. And then the third process, of the authority make a decision. Without science, without the public, any decision, I don't think it will work. Next slide, please. Because of time, I don't want to go this. So early and active case detection, very important, especially in the community level. Community level, early and active case detection is the key. So this is the advantage. You can do this in China because we have a very good community level service, public health and clinical. Next slide, please. And also, web-based reporting system. This one also is published for many, many years and is organized by China CDC. I give you this list. You have a notifiable individual case information system, public health event information management system. All these systems, if you include the death monitoring system, is set in China CDC. Next slide, please. Of course, case reporting requirement. So this is what we, all this from our protocol. Next slide, please. And the case and contact detection and managing the workflow. These are the workflow, especially for those suspected cases and for those close contact. So you have all this flow. This flow is understood by everyone. This is what I mean, but especially the community level supervisors. As good supervisor, the community level is very, very important. Next slide, please. And then case management, as I mentioned already, suspected cases, we need to get them isolated in a single room, and confirmed cases and asymptomatic infection, as you know, and also could be isolated in the same room because they already know it's positive, and also it's monitored for 14 days. That's next slide, please. And case management, also 14 days twice for the nucleic acid the testing, and more importantly, I mentioned community level supervisor to check, make the phone call to any of those cases to make sure they are okay. If they have any symptoms, they will test them again. Next slide, please. So this is what we are doing, contact definition. Because of what we found, asymptomatic infection, they would set the virus 2 days before the illness of onset. This is why we define the contact definition, 2 days before illness onset, very, very important. Next slide, please. And multiple department cooperation, I already mentioned this, I will move further. Next slide, please. And also, I mentioned about 14 days and twice a day for this. Next slide, please. Here you go. So as you know, in the world, we have been talking about mask wearing for a long time. But in our opinion, because it's droplet, transmitted by droplet, you never know whether or not you are infected because asymptomatic infection. So this is why we think mask wearing is very important. You look at Japan, Singapore, and South Korea. So we all wear the mask. Next slide, please. And here we printed out a lot of posters and booklets to disseminate to the public and try to make sure that everybody understand that. Next slide, please. And the health guide, and also the videos we distribute to everywhere. Next slide, please. And also for the multimedia interactive dissemination. So this is why from the very beginning we think youth pandemic is equally important as epidemic. Next slide, please. Of course, we did all these mathematical models. Next slide, please. Now, so we call it we try to define the 4 defense lines in China from the very beginning. Wuhan, and Wuhan, the province Wuhan, and Wuhan surrounding areas, and also nationwide. So this is what we have done. Next slide, please. Now we have to resume, like everybody. I think Jerry mentioned, and also Tony mentioned. We have to now come back, resume the business, resume the social activities. Now we got to dance with the virus. There's no choice. So this is what we start for this. But the most important thing for China at the moment, control the imported cases. We have 2-digit numbers always at the moment for the imported cases. Next slide, please. Yeah. And for all those imported cases, we also developed our own protocols. Next slide, please. Also in the 14-day and twice. And now we try to introduce a health code and travel card. Those who are from the some areas, very serious places, they usually have the code and travel. We know where they are from, and also we have the guidance for the work and enterprises for their beginning. They know where their workers come from. Next slide, please. And also we learned all this. We call this transition stage. We also, again, adhere to the 4 early measures I mentioned to you already. Next slide, please. I think I'll have the last slide. So here you go. So it is where we are in China. We reopen our business. Of course, the school are not open yet. We are planning to have the school open. We are also working very hard for the vaccine, the specific drugs, also the traditional Chinese herbs, and also plays a very important role. That's it. Thank you.
Thank you, George. Lots of things we can learn, and lots of questions, I'm sure, from certainly myself and the panelists and audience. So we'll come back to you. So next we go to Susan Wise. Susan, you've been a leading virologist. You've been studying coronavirus all your life. Tell us a little more about this novel virus, transmission to human, mechanism of pathogenicity. Coronavirus, actually, I believe there are 4 coronavirus that causes common cold. So how is this different, this COVID-2 different from the others? And then, of course, I know you're thinking about therapeutic options, making antivirals. So, Susan, we look forward to hearing from you.
Okay, thank you, Victor. It's really an honor to be invited to be part of this panel. So I'm going to talk about coronavirus biology a little bit off in a different direction from the rest of the talks. And I'm going to try to point out places in the virus life cycle that might be potential targets for antiviral therapies, particularly targets that might be applicable to many, be effective against many, if not all, coronaviruses. So here's the virus particle. It's actually very simple. The inner part is the capsid composed of a very long RNA molecule. It's got the longest RNA genome among viruses. And this RNA genome is complex with nucleocapsid protein to form a helical structure that's shown in the green here inside of the virus particle. And around the nucleocapsid is a membrane that's derived from the host cell. And in that membrane, there are 3 proteins in all coronaviruses. The spike protein that everybody's heard about that gives it this sort of corona or crown-like morphology. This protein the spike is important for viral entry, for binding to receptor and fusion, for viral tropism, for immune response, and for virulence. The other 2 proteins in the viral membrane are this membrane protein M and small membrane protein. And although so much attention is paid on the spike protein, all of these other proteins play really important roles in replication as well. Here's the human coronavirus genome structure, and I've drawn structures for genomes for all the lineages that contain human viruses. All coronaviruses have a very long replicase protein shown here in the purple, and that encodes 16 non-structural proteins that I'll talk about in more detail later. And then downstream are the spike, E, M, and N genes, always in the same order as shown here. And then these accessory proteins, which are the numbered open reading frames here, 4A, 4B, 5A, those are different for each lineage of coronavirus. And that's what really distinguishes their genome organization. So within these different categories, the alpha coronaviruses contain 229E and NL63. Those are 2 less pathogenic viruses. 229E is a common cold virus. NL63 causes croup and bronchiolitis. Then among the beta coronaviruses, there are 3 lineages containing human viruses. Lineage A contains OC43, another cold virus. HKU1 causes pneumonia. And then lineage B contains SARS and the bat-like SARS coronaviruses. Lineage C, MERS coronavirus, and also the bat-MERS-like viruses. So as Dr. Gao said, the sequence for SARS-CoV-2 was revealed very quickly after the virus emerged, and it was shown to be very similar to SARS-CoV-1, and therefore a lineage B coronavirus, and called SARS-CoV-2 because of its close genomic sequence resemblance to SARS. Here's a model of a coronavirus replication cycle, and I drew this in 2005 after the SARS epidemic. This is a simple, very simplified version of replication. We know a lot more now, but just to go through it quickly, the coronavirus binds to its receptor. This is really for any coronavirus. And then it's internalized into the cell by a fusion event with the cell membrane. The viral genome is translated into a replicase, which forms a complex and then produces a lot more genome RNA as well as messenger RNAs, many 6 or 7, depending on the virus, messenger RNAs, all of which produce viral proteins shown here. The viral proteins, the membrane proteins, the M, N, and E, assemble on intracellular membranes shown here. The nucleocapsid protein and the viral RNA complex and form this helical capsid structure, which then buds into intracytoplasmic membranes between the endoplasmic reticulum and Golgi. And the mature virus particles are now in vesicles in which they're transported to the cell membrane and released from the cell, shown here. So I want to talk a little bit about two steps in this replication cycle in more detail: entry and the replicase proteins. Both of these are have been used and are good targets for antiviral therapies. So first, viral entry. Viral entry can occur by two routes into the cell. The first route is the endosomal route. In this route, the viral spike protein attaches to ACE2. This is in the case of SARS or SARS-CoV-2. The ACE2 receptor angiotensin converting enzyme that we've heard about before. The virus is then internalized, it's endocytosed into an endosome, into a vesicle. The next thing that happens is the spike protein mediates a fusion event between the viral membrane and the host membrane, releasing the nucleocapsid into the cytoplasm here to begin the infection. Now, before this can occur, the viral spike protein has to be cleaved, to be activated to cause this fusion event. And in the case of this pathway, that activation often occurs by cathepsin. And this protease requires the low pH environment of the endosome to be active. The other pathway is the direct plasma membrane route. And in this pathway, the virus attaches to its receptor, and again has to undergo a proteolytic cleavage, this time by TIMPers, which people have heard a lot about TIMPers, I think. But it also could be an enzyme like furin. People have heard about furin and the furin site on the SARS-2 coronavirus spike protein. So one of these proteins, one or both of these proteins will cleave the spike here, allowing it to mediate a direct virus cell membrane fusion event, and infection again ensues. So it's what determines which pathway the virus used to enter is a combination of the protease sites, the sequence of the viral spike protein, as well as the proteases available on the host cell, which it's going to infect. And that can vary from cell type and can vary among viruses. Some coronaviruses use each route. Some of them use both routes. And this is really important in thinking about antiviral therapies because, for example, hydrochloroquine would be expected only to work against entry by this pathway because it raises the pH of the endosome. Other inhibitors may do the same, or a cathepsin inhibitor, which inhibits this enzyme, may also inhibit this pathway. However, to inhibit this pathway, you might need a TIMPers inhibitor, for example, Camostat, or a furin inhibitor, or a combination of both. So it's just important to know, or to perhaps be targeting both pathways and really trying to prevent viral entry. Okay, so translation of coronavirus proteins. These are the coronavirus RNAs. This is the genome RNA, and all the blue is RNA. And these are the subgenomic RNAs. This is just for mouse hepatitis virus, but it's very similar for all the coronaviruses. And so these RNAs are synthesized by a fairly complex mechanism in which a negative strand RNA is transcribed from the genome. And then several subgenomic negative strand RNAs are transcribed as well. And these are templates for amplifying more genome and more RNAs, or messenger RNAs shown here. It also involves a mechanism whereby there's a non-continuous transcription so that each one of these messenger RNAs has a leader sequence that comes from the 5' end of the genome, shown here. And all of this has to and then also the RNAs have to be capped and methylated at the 5' end and polyadenylated. And all of this has to happen in the cytoplasm and therefore by viral enzymes. So this region of the genome here, or the replicase gene, encodes 16 non-structural proteins. And these are the proteins that carry out all these processes of RNA synthesis and also process these precursor proteins into mature polypeptides. So what controls whether there are two very long polypeptides translated from this region, polypeptide 1A and polyprotein 1A, 1B. And what controls the ratio of synthesis of these two proteins is the so-called translational frameshift sequence right at the end of polypeptide 1A. And so the ratio of these two proteins is quite important, because studies show that if you do something to alter this frameshift sequence and you make too much or too little of one of these proteins, the virus is vastly attenuated. So this is important. And I want to talk now just about the proteins encoded in the PP1A and PP1AB. So here's a picture again of open reading frame 1A and 1B, and these are all non-structural proteins. There are 16 of them encoded by all coronaviruses, and I want to stress this is conserved among all these viruses. So some of these have already been cited as targets for antivirals. Well, I should just say, let me tell you what the proteins are first. There's a papain-like protease encoded here, and it processes, it cleaves these two proteins from these polyproteins. Then the 3C-like protease is the main protease. You probably heard that term. It processes all the rest of these proteins. And then there's the RNA-dependent RNA polymerase, or the RDRP, and this replicates genome RNA and transcribes messenger RNAs. Remdesivir is targeting this enzyme. This is repurposed from Ebola, as Dr. Fauci told you. And lopinavir and ritonavir are two HIV inhibitors that have been used to target the 3C-like protease, a chymotrypsin-like enzyme. And as Dr. Fauci said, none of these, all of these inhibitors that are repurposed are in clinical trials now, and we don't really know how well they're going to work. So we have proteases that process replicase proteins and an RDRP that replicates genome and transcribes mRNAs. But there are a lot more enzymes and other interesting proteins encoded in this gene. I should mention these people here have been working on protease on SARS or coronavirus inhibitors even before, probably since the first SARS epidemic and MERS. They have continued to work on antivirals for these viruses and continue to do so now. So I just wanted to acknowledge them, colleagues. Okay, so additional conserved coronavirus proteins. There are also a helicase and a primase that are required for RNA replication. There's also several enzymes, these capping and methylation enzymes that are required for capping and methylating the messenger RNAs, which is required for translating them effectively. There's also a protein called XON, or a proofreading protein, to prevent to keep down the error rate during replication. There's also an endo-U that is required for antagonizing the host cell innate immune response. And in addition, there are even two more proteins here, NSP1 and NSP3, that encode several host antagonist activities. So this virus really has a lot of targets that could be used for antivirals. So in summary, there are enzymes that promote synthesis and stability of viral RNAs, capping of 5' ends of messenger RNAs, and protection from host cell sensors and from interferon response. And just to show you one example, I want to just tell you about one example of one inhibitor that inhibits the NSP13, or the helicase protein. This shows molecular models of the helicases of SARS, MERS, and MHV, and you can see that this compound, SSYA10-001, fits into the binding pocket of each one of these proteins. And down at the bottom, we can see that it also is able to inhibit replication of each of these. This is in cell culture. And you can see here that the viral replication, which is read off shown on the y-axis here, decreases as a function of increased concentration of treatment with inhibitor. So this is just one kind of example of an inhibitor that can be useful against multiple coronaviruses. And just to summarize, there are many conserved features of coronaviruses. The entry pathways are host protease dependent. Subgenomic RNAs are synthesized by non-continuous RNA synthesis that could probably be interfered with. The open reading frame 1A, 1B frame shifting and translation is something that needs to be just the way it is. And several viral proteases, the RNA polymerase and RNA modifying enzymes, and really further host response antagonist activities. And I will stop here.
Richard, you're the last person, and we look forward to a robust conversation, so we don't have a lot of time.
Sure.
Tell us about vaccine, because we all set the turning point, and then what happened with this vaccine. Now, you work in this area. Bada?
Great. Thank you, Victor. And
Okay.
It is an honor to be on the panel and to address the National Academy of Sciences meeting. CEPI is an organization that many on the who are listening may not have heard of. It is the Coalition for Epidemic Preparedness Innovations. It was set up in 2017. Our mission to accelerate the development of vaccines and to ensure access for the populations that need them. If we go to the next slide. We just a few weeks ago published a paper in one of the Nature Reviews journals looking at the global landscape for vaccines. And I wanted to just show you a few slides about what that landscape is. When we published the paper, which was on April 9th, there were 115 vaccine programs that we had heard about. As of today, there are 137. Of the 137, we've been able to confirm by direct contact or through the Bill and Melinda Gates Foundation or via WHO that 107 of those were active, and those are the programs that I will talk about. You can see from this slide that these vaccines use many different approaches to vaccine development. So we have a very diverse array of candidates globally. Currently, we've had one vaccine that has been approved for phase 2 clinical trials. That's the CanSino vaccine in China. A number of vaccines now in phase 1 clinical trials, and many others speeding towards phase 1 clinical trials. Next slide. Also just wanted to talk briefly about the geographic distribution of those vaccines. Of the 107 confirmed vaccines, a little under half are based in North America, about 40 in China, Asia, and Australia, and 23 that we know of in Europe, the majority coming from the private sector, a few public sector and academic programs as well. Next slide. So CEPI has evaluated a little under half of the vaccine candidates that we know about through an open call for proposals. We've looked at over 50 vaccine candidates. We've put a portfolio together. Our criteria for establishing that portfolio revolved around 3 strategic priorities: speed, scale, and access. We obviously want to develop vaccines as quickly as we possibly can, but we ultimately need to scale those vaccines to hundreds of millions or even billions of doses to meet global requirements. And we want to make sure that the world has access to those vaccines and that the access to vaccines is not just concentrated in a few nations. If you go to the next slide, this is the current CEPI portfolio. We have 8 candidates that we've announced. We have 2 more that we'll be announcing very shortly. Just to highlight that we have deliberately constructed a diverse portfolio. We have vaccine candidates across many platforms: nucleic acid, recombinant protein, and virally vectored approaches to vaccine development. We have a diversity of partners and a diversity of geographic locations. We'll have 2 partners in China, 1 partner in Australia, a number in the US, number in Europe. We also have a diversity of scalability for some of most of the programs that we're supporting. Ultimately, we'll be able to scale to large doses of numbers. There is an error on the slide. The Oxford program we now assess as having high scalability. Next slide, please. This is a slide from the New England Journal of Medicine article that we published on the 30th of March, developing COVID-19 vaccines at pandemic speed. This is just a very simple representation to show how we intend to accelerate or compress the vaccine development timelines. If we were developing a normal commercial vaccine, obviously we would take it in a stepwise process, and we would accumulate data through the preclinical and clinical development programs, and we would stage our financial risks in terms of the very large investments required to develop the manufacturing process and then scale that manufacturing process up to commercial scale. What we are trying to do to speed the development of vaccines is obviously to compress the clinical development timelines without, and I really want to underscore this, without cutting corners and without taking, without doing anything to undermine demonstrating clearly the effectiveness of the vaccine and demonstrating with certainty the safety of the vaccine. Where we can also compress those timelines, though, is by taking financial risk, by scaling up the manufacturing as quickly as we can, and then beginning to build inventory even while we wait for the data to accumulate as to whether the vaccines work. That means we will be building inventory for some candidates where the data comes in and suggests that the vaccines don't work. That will increase the cost of the program, but if we want to deliver vaccine at scale within our stipulated target of 12 to 18 months from the initiation of the program, we're going to have to be comfortable with those risks, and donors and those who will be paying for the vaccine have to be comfortable with those risks. If we go to the next slide, I just want to say there are a number of challenges ahead. We need to mobilize resources. If our intent is to deliver billions of doses of vaccine in coming years, that is an extremely expensive proposition. This will cost ultimately, not this year, but over the next several years, tens of billions of dollars. And I think the donors need to understand that. I think they also need to weigh that cost against the cost of the pandemic, which is costing hundreds of billions of dollars to the global economy every month. So if we shorten the pandemic by a month, we're saving hundreds of billions of dollars. And that's the calculus that elected leaders need to make as they think about funding these programs. We must address liability and indemnification. Concerns about this have slowed down accelerated vaccine programs in the past, going all the way back to the efforts to develop a vaccine against swine flu in 1976. They slowed the delivery of vaccine during the 2009 pandemic to developing countries. We must address these fast. We must address them prospectively. I think obviously we need to ensure equitable access. For the interest of time, I won't go into that. I am concerned about what I've called vaccine nationalism, and that is the tension between obligations that elected leaders will feel to protect the lives of their citizens versus the most efficient way to protect their livelihoods, which is to make sure that the epidemic is addressed globally. Finally, we need to make sure that countries globally are ready to deliver the vaccines that we develop, that they have the cold chains in place, that they are ready to undertake massive vaccination programs, and that we have worked out prioritization schemes. If you go to my last slide, I just want to conclude by talking about something that was announced yesterday. This is the ACT Accelerator, which is a global collaboration to accelerate the development, production, and equitable access to diagnostics, therapeutics, and vaccines. The organizations at the bottom of the slide have all banded together, and we have banded together with willing private sector partners in an effort to collaboratively speed the development of countermeasures and to ensure their access to the world. This will be, as Jeremy said at the beginning, this will be our global exit strategy from the pandemic, and we will need to do this together. Thank you.
Thank you very, very much. Thank you, everyone, for outstanding presentations. So in the interest of time, I've clustered our conversation to 3 or 4 categories: mitigation, testing, treatment, and vaccine. But let me start with vaccine first, because Tony Fauci at NIH is involved with a program called ACTIVE. Others involved with a program in working with industry. Jeremy, you, and to some extent myself, been involved with this ACT, if you will, ACT and ACTIVE. So let's talk about how all these things fit together, and particularly how do we assure that people who don't have the resources get access to vaccine? Both, first of all, in low-income countries, how do you assure equitable access? And then how about in our country? We talk about vulnerable populations. So let me start off with maybe Jeremy, Tony, Richard, and Sanjay. Let's just have a conversation. You don't have to be worried too much about etiquette or courtesy. Just go and start talking. Tell us about.
Well, I'll jump in. I'll say one thing that is very critical to understand about the problem of access in a pandemic, which I think is different and very importantly different from some of the other access challenges that we've tried to tackle in global health. Those other access challenges are usually access challenges that are created by a price problem. And the problem in the pandemic, at least initially, is going to be a scarcity problem. And ultimately, over time, obviously, we want to turn that scarcity problem into a more traditional access problem if we can. But the critical issue at the beginning is just to make as much stuff as we can that we're sure is safe and effective, and then to move it to the people who need it.
So, Victor, let me jump into it. Unfortunately, as I mentioned to you before, I have to leave in a short while because I have to get down to the task force meeting at the White House. But I think one of the things that has to do with both access and success with a vaccine is the fact that you need more shots on goal for a chance of getting a safe and effective vaccine. But if you have all of your shots on goal go into the net, then you got a lot of other companies that are making vaccines. We can't just have one vaccine. I mean, our program at NIH, we've developed a vaccine and likely more than one ourselves and are partnering with industry. But we are also supporting directly or indirectly, mostly through the access to our global network of clinical trial programs, a number of other pharmaceutical companies who are the primary movers for a variety of vaccines, others including Sanofi and J&J and others, so that what we really do want is we want multiple successful vaccines, because you're never going to get to, I mean, I like the slide that Richard mentioned, which was, speed, scale up and access. And I think the only way you're going to do speed, scale up and access is to do a lot of shots on goal and a lot of goals from those shots. So we're supporting all of that. Obviously, our first obligation is to the United States, but we're very sensitive to the fact that as a country that has a big research and development operation, that we do have a global responsibility. And that's why we'll be working, with CEPI and with others to see if we can help in that regard. I'll stop there.
Thank you. Jeremy, comment?
Yeah, only to say, I mean, these various initiatives, whether linked to the work that's going on in China with George is very involved in, or indeed the works with Tony over at NIH. At a scientific level, these are all linked up. And that's crucial, because we don't know where the best treatment or the best vaccines are going to come from. They may be developed in the usual countries that have driven innovation and research and development over the last 20, 30 years. They may come from unexpected places. And the critical element is not where they come from, but do we share them when they are available?
Yes.
And do we then commit to making the manufacturing and the distribution work? And that's at the heart of the scientific endeavor, whether it's at NIH in the United States, it's at China CDC, or indeed it's in CEPI and the ACT program that was announced yesterday. And, as scientists, we need to commit to that. We do live in a tense world, and we live in a slightly polarized world. Yeah. And I think our job as scientists is to bring people together.
Yeah, to save time, I would simply say that it looks as if industry is also agreeing to do this together. And that's really, really important. And the fact that governments are coming together to say, we'll de-risk industry, we'll work together, we have one program, is so important. So all three entities, we say, China, US, and this global initiative is working together. So thank you very much. So in interest of time, Tony, before you leave, let's talk about testing and mitigation, because this is the preoccupation of our country now, talking about this. And of course, I want to ask George the question too, because you're saying you're sending people back, opening the school again, etc. In our country, a lot is dependent on testing. And so it's really important to look at what is testing, what's our strategy, and and how many do we need to do in order to know that we actually have a good feel. For example, right now, our positive rate for serological testing is about 22%. say you need to go down to, actually, no, the molecular testing. Here needs to say you need to go down to less than 10%, WHO. So let me turn to all of you, particularly Tony and George, and maybe Sanjay, talk about testing. How many do we need?
Right.
What kind of testing, and how does it fit into the strategy overall of the mitigation and then relaxation?
Yeah, let me take a very quick shot at it first, Victor. Well, I think it's a bit misleading to when you say how many tests do you need. You need enough tests so that when you are doing what we're trying to do right now, which is try and ease our way back, that you can very easily identify, test, isolate, contact trace, and as George says, get those who are infected out of society so that they don't infect others. I mean, obviously, if you test and you have a high percentage of people, you're not having enough tests. You really need to get it so that you have maybe less than 10% as a quantitative number. But we don't want to get fixated on how many tests you need, but do you have enough tests to respond to the outbreaks that will inevitably occur as you try and ease your way back into the different phases? So right now, we're doing about 1.52 million per week. We probably should get up to twice that as we get into the next several weeks, and I think we will. Testing is an important part of what we're doing, but is not the only part. But no doubt it is important to be able to do the identification, isolation, and contact tracing. There's antibody testing, which you can get into later, that's totally different than testing for the virus. I'll stop there.
Okay. George?
Yeah, let me put it in a way. When I learned from America, when I was in the USA, I learned something like when you buy a house, location, location, and location. To prevent COVID-19, testing, and testing. I think that's quite understandable. So you try to test as many as you need to do. So look at what happened in China and also in Korea. Korea even set up the driving through testing, try to isolate any suspected or close contacts or any places. So testing as many as it should be. Thank you.
Sanjay, I think it's more complicated, isn't it? Because it's who you test, where you test, and as Tony said, what's the strategy afterwards? That is, if you test fewer, but maybe specific populations, and you do contact tracing, it's different from testing the entire population. So there's a range of people saying we need to do 700,000 cases a day, or tests a day, to 2 million. Scalability is another issue. So while it's fine in a kind of a philosophical issue to say we test as many as we want, I think we do need a strategy of what's your priority? How do you put it out? How do you use the cases that you can to trace and quickly do contact tracing? And so that's the question. And given the size of our country, I think that's the challenge we're faced with. Sanjay, what are your thoughts?
Yeah, no, I think absolutely, and I think initially being able to explain what the role of surveillance is, and how it's going to be done, I think is really critical. And I think what the task force has talked about is that at first at least we're going to do surveillance in areas where we think nursing homes. Places where people can have a hard time physically distancing themselves, to get an idea there, but then to really look at the general population, and you need much broader testing. I think what is interesting as some of these places think about reopening now, what is the practicality of testing in these places? When we say we want to have widespread testing, part of that is to get a better idea of just how far this virus has penetrated, but also to start to give people some confidence, I think, in returning, starting to reopen these economies. I live in a state now where things opened up yesterday. I think there's not enough testing here. It doesn't meet this 10% sort of positive sort of rate that Tony was talking about. So how do we explain that to people? And not just point of care testing, but ultimately point of location for work or for these businesses. Are people going to be tested on a more regular basis as they start to enter into the workforce? Are they going to be tested once a week? Are they going to be tested twice a week. And I think one of the questions that comes up is, we talk a lot about incubation period, but now as we know about both asymptomatic and pre-symptomatic spread, from point of origin to point of contagiousness or infectiousness, what is that latency time? What is that, time period? Because I think that's going to be something people want to know in terms of frequency of testing. I don't know how long that's going to last. I mean, I guess it's predicated on a lot of things that Dr. Hatchett was talking about, but I think that's something that we have been talking quite a bit about as we do these stories.
George, do you want to come in? George?
Sorry, it worked?
Yes.
Yeah, I think, at the moment in China, we try to resume all the business. We try to, set up all these tests from the hospital to the local CDC and also in the community level. We just test as many as possible. Thank you.
Well, so my only point is we do need a strategy. Given the size of a country like ours, and China is even bigger, you need some kind of strategy, as Sanjay talked about, whether you're looking at high-risk population first, you look at communities, and those that you test, you can do contact tracing rather than try to test everybody, right? Because it's not scalable. And we won't have time to go long. I'm told we can go over by 10 minutes if you're willing. The serological test is another big issue. Let's talk about immunity. And whether Susan or Jeremy or anyone talk about herd immunity, and then the issue about measuring your antibody, IgG, IgM, and to know how much immunity there is in the population, assuming that measurement reflects immunity. So can someone say a word about that?
Susan, coming in?
Well, Jeremy, why don't you go first?
Just a very quick comment on the testing. The testing has to be driven by the epidemiology that we know about and the epidemiology we can anticipate. In Europe at the moment, there are really three overlapping epidemics going on. One in the community, one in care homes and places where it's difficult to distance, including prisons, and one in hospitals. And the testing has to meet those three overlapping epidemics, because if we leave either one of those out of the question for testing, then they will seed the other ones in the future. And so the testing can't just be blanket. There is not the scale in any country to do that. It does have to be smart. It does have to be adaptive. It does have to be driven by the epidemiology. On the antibody testing, for which there's been great controversy, I think certainly in the UK, it looks as if by about a month or 6 weeks, most people have measurable antibodies. That is good news. Whether they are neutralizing antibodies, whether they'll protect and give immunity over the long time, we don't know yet. Whether that immunity will be transmission blocking or will stop severe disease, we don't know yet. And these basic science studies, in a way, are absolutely critical going forward. Also, absolutely critical is that when we do implement testing at a country level, that we know those tests work. Because if the tests don't work, particularly I'm thinking now about the antibody test, if they're not specific, if they're not sensitive, and they don't have good negative and positive predictive values, they will at best be useless, and at worst they'll be dangerous. And so before we roll them out, we've got to be absolutely sure they work, and we're absolutely sure we know what they're telling us. Otherwise, we will make bad policy decisions on bad data.
That's quite right, Jeremy. We do have a problem, at least in our country, that we're flooded with tests which are not FDA approved, but they are actually available for distribution. And the false positive or specificity can be huge. Yeah. Imagine that you had a coronavirus infection of a common cold. You have the immunoglobulin, and you can't tell one from the other, right? So, Susan, talk a little bit about the biology of that side.
Okay. The only thing I was going to say about that is I really doubt that any of the other coronaviruses are going to confuse the antibody test, because they're really different. I think the only one that could possibly cross-react would be SARS itself. And I wonder if people that recovered from SARS, maybe George knows, have people looked at whether they have neutralizing antibodies against this virus or any kind of response against SARS-CoV-2?
I think there's no data, because we do and also from the current data, a lot of publications, including some from my colleagues, looks like all the new monoclonal antibodies, best monoclonal antibody against SARS, it doesn't work for SARS-2.
Does not.
So
Does not.
there looks like no cross-reaction.
It seems like there should be some, no?
Yeah.
Yeah.
I think there is, Susan, at least some reports actually from Canada that suggest there is some neutralizing activity. But the other thing, Victor, we have to worry about, which is not quite a theoretical risk, we do also have to just be concerned that antibodies are enhancing. And that is critical immunological and epidemiological data that we must gain urgently to make sure that when you've had one infection, you're not more at risk of having a second. That's going to be critical to vaccine development. Actually, the evidence I've seen suggests there is not enhancement, but it's something we will have to watch out for.
Thank you. Let's talk about treatment for the remaining 5 minutes, and whether I can pose it to anybody, maybe Richard or Susan, who knows the virus, or Sanjay and others. To my knowledge, there's 7 drugs, 4 repurposed, 2 new, and 1 adjuvant. There are about 250 trials everywhere, in China, everywhere. So maybe the questions are what to expect, and is there good coordination, and particularly when think about all the harmonization clinical trials protocols, the question of design, and then ultimately, of course, as Tony said, we do need evidence through randomized controlled trials. So perhaps someone can just bring us today, what are the drugs being studied and how are they studied and what do you see in the future? Richard, you want to start first?
Actually, Victor, I mean, we're focused exclusively on vaccines. I'll make one very quick observation and then turn it over perhaps to Susan or Jeremy, who could talk about the therapeutics accelerator that Wellcome and Gates have established. I mean, my one general observation is that we should be realistic about our expectations of antiviral therapy. Antivirals are likely to help. They'll need to be used early, but they probably won't be a silver bullet. And that's our experience with previous efforts to develop antivirals against other drugs. Critically important to do it. I think we're all hoping perhaps the monoclonal antibodies, when they become available, will be efficacious, but we need to be tempered in our expectations. Over.
Susan?
The only thing I was going to say, from what I understand from the clinicians, that and from the samples that we've looked at in our own lab of specimens from patients, that the antivirals would only really be effective very early in the infection. By the time people are sick are very sick in the hospital, there's not so much virus coming, at least in the nose. So that antivirals should, if possible, I think that's a problem because we don't really know when people are infected, that's the time when they need them, when we're not sure. So I think they need an antiviral early and then an anti-inflammatory late, and maybe that combination will do until there's, but I agree also a vaccine is really going to be the answer.
Jeremy, some thoughts?
Yeah, I think we need to see therapeutics both in terms of prevention and in terms of treatment, whether we have drugs or monoclonal antibodies that could prevent getting people being infected. We don't yet know if chloroquine would work. I would guess it would more likely work in a preventative strategy than in a treatment strategy. Richard's point about the track record of producing antiviral or immune modulating drugs in acute viral infections has not been great. And I think part of that is because we have to treat early in most antiviral drugs, and then we don't really understand the very fast dynamic changes in the immune response that are going on over the course of what is only a week or two of illness. I suspect at some points you want to increase that immune response, and at other points you may want to suppress it. And getting that right within that very tight dynamic is extraordinarily difficult. And, that speaks to evidence from influenza, from dengue, from yellow fever, and many others. So I suspect in the end combinations will be needed, but getting the timing right of those combinations, a huge challenge.
There is another angle to this. COVID-19 patients have cytokine storms, which actually, you hear story after story about what that means and how devastating that is. And there are people working on interleukin antagonists and others. So does anyone want to talk about that? Sanjay, do you know much about this? Sanjay?
Yep, I'm sorry, unmuted myself. I think I better pass that question on to somebody else, specifically about the immune modulators. One thing I'll just say, it's interesting, just picking up a little bit on what Dr. Weiss was saying. The data that we look at seems primarily, we're looking at outcome data, and in the, it's very few of the studies that we've seen, at least, have been randomized. So seems maybe some of these patients are getting some of these medications early or even prophylactically. Many are getting them with more severe or even critical stages of disease. With the early or prophylactic, it becomes very hard, it seems, to look at those studies in a meaningful way, because so many of those patients seem to would have recovered likely anyway. So, to really understand the impact of these medications early on, it seems it's hard to power up those studies enough and to get meaningful results. I don't know if you agree with that, or how to better present that, but is that something that we should be looking at differently than just outcomes data, actually looking at, a viral load or something else within these patients? But I think it seems like a question mark right now. As Jeremy was saying, it doesn't seem like there's something that we can present as promising, even really. The remdesivir data that was posted on the WHO website briefly a couple of days ago did not look very promising either. Some, I guess, 300 patients or whatever. But anyways, that is one of the issues that I think comes up a little bit. But regarding the immune modulators, I better pass, on that. Okay. Somebody who knows more.
I think Jeremy's about to jump right.
So there's a whole pile of them, Sanjay, being developed. Anti-IL-6, IL-10. I mean, you name a immunokine at the moment, and there isn't a attempt to look in vitro in animal models, or sometimes in clinically. Getting the combination rights of those, I think, will be a great challenge. But just to your point there about scale, if we're fast enough, and there's a challenge to that, there are millions of people with this infection, and there will continue to be millions of people with this infection in the weeks and months ahead. So there is no excuse really for not conducting very large, well-designed, randomized controlled trials to give us the definitive answer of what works. And if we miss the opportunity in this first wave in many countries, and we're not in a position when the second wave comes to know what is bad, what might be good, and what is good, then I think as scientists, we should take responsibility for not having gained that data.
Thank you. A lot of pressure on us. George.
Yeah, can I make two more points for that?
Yes, sure.
The first, when you are talking about cytokine storm, looks like one paper, I think is coming out from the PNAS, from the proceedings, looks like tocilizumab, the targeting for the IL-6 receptor, it does work. I think they have some data from China here. And the second point I want to make, because the virus lasts for a long time, looks like convalescent plasma also works, because when you have very severe cases, those things, severe or critical, it lasts for a long time because the virus lasts in the lung for a while. So convalescent plasma also works. Thank you. Over.
Thank you for pointing that out. I was going to bring that on. So let me close this meeting by asking you guys this question. And this, of course, haunts many of us who work in global health. It's called lessons learned. I mean, how many times have we said lessons learned? Many. I know everyone on this panel, and certainly national academies, Wellcome Trust, GPMB, all written about what's needed and what lessons learned. And yet we keep on finding ourselves in the same situation. And I'll be interested in hearing, is this different this time? And it's not only preparedness for the next outbreak, but it's also an issue of how a society is going to come together and learn how to work together to make a better society. Then finally, I think we have all adapted to so many things that we thought we'd never stop doing, and will our society be very different in education, remote learning, and so many other things. I'll just ask each one of you to say just words and just go around the table, starting perhaps first with Susan. I'll just go down this way.
Well, I do think that I think that we're going to learn more of a lesson from this one than we did from SARS and MERS, because of the amplitude of the spread and the problem. I don't think it's going to, I mean, with the other two epidemics, it seemed, at least the science world seemed to forget about it pretty quickly, and not, we never really finished making those vaccines that I think a lot of money was spent on starting them. They never were actually tested, I guess. Other people would know that better than I. And as far as how our world's going to change, I think it's going to be different. But I really miss going to work every day and seeing people. I really miss going to my lab and hanging out. So I hope.
Thank you, Richard.
As the first thing I've got to say, Victor, is as the father of 3 school-age kids, I want them to go back to school. We are not doing great with homeschooling, but that's beside the point. Just one really quick observation. I think COVID represents an absolute watershed. The impact on global society, on the economy, on literally everybody on the planet is going to absolutely change the approach to preparing for these kinds of events in the future. It will be a very, very different world.
Yes, but Richard, you know the issues. First, are governments willing to invest in something that may or may not happen? Second, are the countries want to work together to prevent something that may or may not happen? Third, is the public willing to give up certain things that may or may not happen? That's always the same issue. So let me go to George, and then Sanjay, and then Jeremy. George?
Okay, 2 points. First, I want to repeat whatever you said. Learn how to work together. That's the lessons or experience we learned from this time. And second, respect our mother nature. Protect the earth by doing good things for the ecology, by doing something for the climate change. We should try to protect our mother nature. Thank you.
Thank you. Sanjay?
There's obviously the broader sort of, I think almost more philosophical issues, but I think, more specifically, I think the idea of how we look at supply chains in the world at a time when everybody on the planet wants the same things, how we're more collaborative about that, I think is an issue that hopefully will be a lesson learned. I think to your point about surge capacity within hospitals, as a clinician, that is something that, we obviously, everybody thought a lot about, but we were thinking about it on a very granular level, how exactly to handle significant surges. Obviously, hospitals, if anything, have gone down in the United States in terms of ICU bed capacity over the last 10 years, not up, despite population growth. And part of that is just driven by economics, Victor, as you mentioned. So how do we invest in those types of things? How do we prove that it's worth it? I do think, as journalists, I think we're a bit cynical about how long people's memories are, but I agree with Richard that I think this is different. This is a watershed sort of moment, and I think it's something that's going to be indelible for some time. I think on a personal level, I think how we interact with each other, how we think about hygiene, whether handshaking, things like that. We're wearing masks now more in this country, something that culturally we hadn't done in the United States. So I don't know whether some of those things will last, in perpetuity, but I do think there's going to be some significant changes here. But I think there's a parallel between medicine to individuals and public health in this regard. I think, we oftentimes tell people to eat right and exercise and live in these healthy ways. And as a result, nothing will happen to you. It's not the most inspiring message, and it's very hard then to prove a negative. I think we're dealing with that same almost, psychological sort of challenge when it comes to pandemic preparedness and the whole pandemic paradox, that if, in the end, if things aren't as bad as some of the models suggested, people will say, well, look, you're fear mongering, you hyped it, whatever it might be. And if they're worse, then, it'll just be that we were under or unprepared. So it's probably hard to get it exactly right. And I think you have to accept that and still try and learn as many lessons from this as possible.
Thank you, Sanjay. Jeremy, you have the last word.
Thanks, Victor. And Victor, thank you on behalf of everybody for pulling us together. It's been a great pleasure working with you over the last few weeks as well, and amazing. Just 3 very quick comments. One is you have to invest in public health. You don't know when you'll need it, but you will need it. And if you don't invest in it, it'll come back to haunt you. Secondly, you have to invest in science. You have to, and that includes social science, basic science, laboratory science, and clinical science. And again, you'll never know when you need it, but you know you will need it. And you cannot do that in waves that come and go. You have to do it permanently. And thirdly, how small the world is. This spread around the world in 60 days. And if you look at the great challenges of the 21st century, epidemics, drug resistance, climate change, they cannot be answered by narrow nationalism. If we don't learn from this, that we've got to work together across borders and across political views, then we will face further crises like this in the future. So coming out of this, we, and it'll be us that own this, we're going to have a choice. Do we go back to a more polarized world, or do we find a better place to be?
Well, thank you. I can't thank all of you enough, really. I mean, George, 3 o'clock in the morning, 3:30 now. Maybe you don't have to go to bed anymore. Just go to work. But every one of you, please, as I say, thank you very much from the bottom of my heart, and thank the audience for sticking with us. It's been, to me, one of the best conferences I've attended. I learned so much. I didn't have to do much. You guys do all the talking. It's just fun. Let's do it again. So let's work together. The last words from every one of you. We need to work together for a better planet and a better society. Thank you.